Welcome to the Hive LiveCell Imaging workshop. We're really excited to get started and to present what we think is the current state of the art in LiveCell imaging, microscopy. Okay, so it's gonna be like held in 3 parts. So first I'll give like a general introduction to LiveCell imaging, very superficially. We'll touch upon a number of techniques that are out there. Then Yang Cheng, who's here, are going to take over and give some background in fluorescence microscopy and some advanced imaging techniques. And finally, after a break, Ashton Francis for tracking and correlation fluorescence, EM correlation microscopy. Okay. So in the interest of time, I'll move on, but so those are like number of applications that you could use LiveCell imaging for and that are going to be difficult to tackle through fixed cell imaging. And so assuming there's no artifact, fixed cell imaging doesn't really reflect the whole dynamics of, thank you. So there's wonderful advantages for LiveCell imaging. However, there's also sort of limitations, and I don't know why. Cells don't used to be exposed to direct sunshine, and actually sort of to extract enough signal from fluorophores. They ordinarily exposed to excitation lights many times over the sunlight. And so that pertains to, leads to phototoxicity and photobleaching, which is a major limitation. So it's also as a result to trying to spare cells from dying and fluorophores for photobleaching. There's limited signal to background ratio usually when you do LiveCell imaging, so that's a compromise. And so you also need, because cells are mobile and there's like some fast processes going on, you need a good temporal resolution to resolve some processes. So those are sort of major limitations of LiveCell imaging. So in general, so there's different ways of doing epifluorescence imaging, and some of them are depicted here. So generally, so you expose cells to excitation light, like here. So in wide field fluorescence, expose cells to excitation light, and then whatever fluorophores emit, and collect that signal and analyze. So in confocal imaging, the out of focus light is being rejected by the spin hole on emission side, and so you could, as a result, do these optical slices and look at different parts of the cell at better resolution. If you need a faster temporal resolution, one could go through spinning disk confocal, which is basically this NIPCAL spinning disk system that creates a pattern of illumination at the same time and rotates it. And it has also reasonable confocality to sort of make several slices here and look at sort of light cells. A better way to sort of selectively expose fluorophores to excitation light would be this total internal reflection microscopy, where excitation light is reflected of the cover slip boundary, and as a result, only like narrow layer of about 100 nanometers at the boundary of cell to cover. So what's not reflected in the slide, and as an add-on, this is a new exciting technique of light sheet microscopy or selective plane illumination microscopy, whereby a light sheet, narrow light sheet of roughly a micron or less in thickness is created, and it could scan the cell and then selectively excite number of fluorophores across the cell as opposed to selective excitation of fluorophores at the basal membrane. And this is really a powerful technique that sort of promised really to revolutionize the field, especially for small animal tissue imaging as well as single cell imaging. So this is how it looks. So you have like excitation objective, an objective that collects light, it goes at the angle, at the right angle, but at the angle to the cell, and this is how one of the system looks like. And so this is like a little trough where cells sit, and one objective is excitation, another for emission light collection. They could switch over as well to create a better isotropic resolution. And this is a movie of light sheet, how it works. You see the selected plane illumination in a cross-section of cell coming at the angle, and then you could really quickly scan it to a cell and get like three-dimensional view of cellular organelles and tissues. So, and that could be done incredibly fast, and it causes the least amount of damage because you only excite fluorophores that you're actually imaging at the time, as opposed to sort of exposing whole cells and all cells in the field as a matter of fact to excitation light. So some of the biological applications I already mentioned. In the interest of time, I won't dwell on this, but again, sort of this number of things that are really exciting, and the, whatever the applications are proper for certain things, such as let's say confocal spinning disk. It could do look at three-dimensional fast processes. There's ways to detect fluorescence recovery after photobleaching, which I'm going to describe in a minute, as well as some of the processes of cell-safe dynamics, organelle movement, etc. So, and for every application, I just put like a simple chart that would guide you to select proper, fluorescence imaging application. Let's say, depending on what you're looking at, if it's like at the basal membrane, then perf microscopy would be, technique of choice. If it's a thicker sample, so several layers of cells, then two-photon microscopy that has deeper penetration into tissue and better conflicality would be the way to go, etc. So, in other words, I mean, there's not a single technique that's ideal depending on what applications you have in mind. You have to select a proper technique and make sure you have access to this. So brief introduction to fluorophores. As you all know, there's a number of genetically encoded fluorescent proteins that have been introduced a while ago, and they essentially revolutionized the field of LiveCell imaging as well as fixed-cell imaging. There's also genetically encoded tags such as halos, SNAP-tag, etc. That could be then labeled as organic fluorophores, but they're genetically encoded target proteins with those little peptides or tags. You can also, there are ways to introduce through orthogonal labeling the non-natural amino acids at selected locations, and they're labeled into quick technology. So that sort of a spectrum of things available. There are organic dyes that could be reacted with the above fluorophores, and also non-specifically to proteins, to amino groups, etc., as well as quantum dots that sort of come up with bioaccumulated or other sort of functionalized means that would allow you to pack your molecules with quantum dots. And the advantage of quantum dots, obviously, they're virtually indestructible, so they can write and could be followed for a long period of time. This is just a simple, poster to show that, sort of from fluorescent protein perspective, there's now a wide spectrum of proteins, starting from blue to near infrared. And you could sort of select them and do multicolor imaging. The limitations of fluorescence proteins are obviously, so again, limited stability, limited brightness, as well as broad emission spectrum and excitation spectrum. So you have the spectral overlap and bleed through. So trying to do multicolor imaging, essentially 3 to 4 color. 4 color would be pretty challenging already because of this limitation of spectral bleed through. So in that regard, hats off to flow cytometry folks for doing like 16 colors and whatnot. I don't know how to do it. So again, this is like a little table to show that, sort of the advantages and disadvantages of different labeling strategies: organic fluorophores, genetically encoded fluorescent proteins, peptides, quantum dots, and sort of indicator probes. But so you could basically pick and choose, but it's important just to optimize the labeling strategy, the expression levels, just to make sure that things are not overexpressed or toxic to cells and don't cause like aberrant processes that you might be watching and then wondering what's going on. So as I mentioned, so the main challenge and limitation of live microscopy is essentially photobleaching and phototoxicity. So, and this slide illustrates that. So you have fluorophore expressed, and after repetitive exposure to light, you see photobleaching. But most importantly, that causes also phototoxicity. So the cell is blooding now. So that also is really an important limitation. So we really have to avoid these things and make sure that cells are happy and we don't really lose signal as shown here. So there's a number of ways to address it, but the most important guidelines are here. So just be very stingy on excitation light. So you have to really go as low as possible with excitation light and really sort of think of your photon budget because fluorophore has a limited number of cycles that or limited number of photons that it could emit. And so you kind of have to budget it to make sure that you could do what you want to do. If you're doing short-term imaging, you could go fast and expose it to, again, minimum light, but quickly. But if you're doing that like hours or like overnight imaging, then you have to really space your excitation light and acquisition so that you could get your cells to emit and sort of stay alive. Excitation emission set needs to be optimized, obviously, for given fluorophores. And again, one has to be very cautious of number of Z-stacks and acquisition frequency, as I mentioned. In that regard, really, this selective plane emission microscopy without the light sheet scanning to the cells by far is the least damaging technique. So if you really want to keep cells alive and do it fast, that's sort of the way to go. Unlike fixed cells, there's really sort of the ways, sort of chemical ways to minimize photobleaching, in my opinion, are not that efficient. There are different oxygen scavenging systems that you could use, including this X-rays, etc. So that those are sort of okay for organic fluorophores. They're not as efficient for fluorescent proteins. And so, arguably, this may or may not help, but one has to kind of optimize the conditions depending on fluorophore and applications. So again, poor signal to noise ratio. That's to the background ratio as a result, because you have to really be very careful, how much excitation light you use. That's sort of a challenge, and you have to optimize, make sure the cells are the fluorescent, etc. So what are the essential components to do light microscopy, obviously, other than microscope? So excitation is generally less important because, as I mentioned, for LiveCell applications, you're not light limited. So you actually have to attenuate the light. So the power is not a consideration. Some of the coherent light sources are better for some applications, but you could go pretty much with any excitation system, depending what you're doing. Highly sensitive detectors are extremely important, and sort of the best detectors out there will give you better yield, such as intensified CCD cameras, etc. Very important to keep cells in focus, especially if you're doing long-term imaging and you have like Z-drift and things go out of focus. So hardware out of focus is incredibly important. There's some systems that have technically sort of contrast-based out of focus, but that entails exposure to excitation light, and that sort of increases photobleaching. So this is important. And there's this number of microscopy chambers, enclosures that sort of need to control humidity, pH, and CO2, obviously to keep cells alive and functioning. So that's actually also very critical to do LiveCell imaging. So this is sort of one way to say, I believe it's a Zeiss system that shows like a full-blown system to really do LiveCell imaging that has this microscope enclosure chamber that maintains temperature, proper humidity, and CO2 control through this module. So you can actually connect it to CO2 tank, and it will maintain 5% CO2 in the chamber and reasonable humidity. And you could do really sort of overnight or multiple day experiments. Actually, what's your record? 3 days now, or something like that. So you could sort of keep them alive for a while. Also important in this regard, you have to have like obviously carbon dioxide in there and sort of bicarb containing medium that kind of maintains proper pH and allows cells to function. As I mentioned, so the excitation sources are less critical. They could be laser, LED, xenon lamp, etc. Very critical are the sort of detection devices, EMCCD, intensified CCD, that now reach in the 90+%, 95% quantum efficiency. So really sensitive detection devices. CMOS cameras are also fast and sensitive. And for laser scanning confocal systems, they are mostly photomultiplier tube-based, but there's like hybrid gas detectors now that are more sensitive, have better quantum yield, and actually will give you better sensitivity for LiveCell imaging. So that's sort of components of the system. Also important, if you if you're on a budget and don't want to use this full-blown system, there are some stage top choices that you could sort of maintain again temperature and perhaps humidity. So there's a number of Peltier-based heating chambers that are compatible with, say, 35mm microscopic dishes, where you can sort of control the temperature, and there's a way to control humidity as well. But if you choose this as opposed to this enclosure that heats the entire system, including the objectives and everything, what have you, this you have to then go with objective heaters to equilibrate temperature across the dish. Otherwise, the objectives are acting as a huge heat sink, and you're not going to have uniform temperature. So there's a number of ways to do this, and then humidity and CO2, there's additional gizmos that you have to kind of hook up and make sure that they can really control what they need to control in terms of homeostasis and cell viability. Hardware autofocus, so like pretty much every major microscopy company now has this sort of a way to maintain focus. And it's sort of universally based, there are fancy names for this, universally based on this IR light reflection from the cover slip. And so from that, there's a way to kind of look at the pattern of reflection and maintain the distance between objective and the cover slip, and you could basically maintain a certain offset from that and keep cells in focus, which is incredibly important to the successful LiveCell imaging. And on top of this, now there are now available, if you're interested in high throughput systems, you could really do this 96 well, high content, high throughput LiveCell imaging that have a number of ways to detect it, wide field, confocal, etc. Streamlined way to image live cells, signaling and whatnot. So then I wanted to dwell on some of the advanced microscopy techniques. So many of you, I'm sure, are familiar with FRAP, fluorescence recovery after photobleaching. And this is a technique widely used to look at protein or molecule mobility, could be proteins, could be lipids, whereby a small area, usually several micron in diameter or size, is photobleached quickly, and then recovery of photobleaching is followed over time. This basically curve from which you could estimate or measure, depending how you do your photobleaching, the diffusion coefficient of molecules and determine their mobile fraction. So partial recovery means there's some immobile fraction of molecules that don't get back and recover. And this has been really important technique to do this, and it's like readily implementable in a number of systems. So that's good, but it's sort of on more microscopic scale. Another widely used technique that everyone is familiar with is FRED, fluorescence energy transfer. And so where you have spectral overlap between donor and acceptor, and due to this overlap, there's non-radiative energy transfer between donor and acceptor, and essentially donor emission as a result is sort of quenched, it doesn't emit as well, and acceptor exhibits sensitized emission. And so there's a number of ways to measure FRED, but it's conventional ways of measuring this are fairly tedious, and it's like intensity based, and you have to have a couple of cubes and do corrections to actually artifact prone because of bleed-through, spectral bleed-through between donor and acceptor. You have to really correct for this, and expression levels of your protein don't affect your accuracy. So in other words, if you overexpress things, it's kind of going to affect FRED efficiency, also dependent on acceptor concentration, etc. So that's not readily compatible with LiveCell imaging. There's also a pretty simple technique of acceptor photobleaching, when you essentially photobleach acceptor and see increase in donor fluorescence. That's actually a nice technique, but also incompatible with LiveCell imaging because this is one-time deal. Photobleach is done. And so lifetime imaging is actually the way to go with LiveCell imaging. So fluorescence lifetime, so every fluorophore has a certain lifetime of emission after it receives excitation photon. And so the lifetime of donor actually is decreased in the presence of acceptor. And so you could measure decrease in the lifetime of donor fluorophore, and which is actually concentration independent and fairly robust. There are two ways to measure lifetime, time domain and frequency domain. In time domain, I'll tell you a little bit about this. You do like a time correlated single photon counting, and then look at essentially measure the lifetime of your fluorophore, and then so with and without acceptor. or frequency domain, which is less sensitive but fast, and you measure the phase shift between your modulated laser fluorescence for excitation and emission. This phase shift is proportional to lifetime. For the time domain, which is a little slower, it's, as I mentioned, this is time correlated single photon counting. So pulse laser, very short pulse, and then you essentially measure the time for photon arrival after your pulse. And as you do that for a number of pulses, you create this sort of a lifetime histogram of decay of fluorescence after pulse. And so based on that, we measure for exponential fit a lifetime of fluorophore, and then changes in lifetime which essentially reflect, in this case, spread or some environment change in fluorophore intensity and in the protein-protein interaction. So this is just an example of essential intensity image, which is less revealing, but fluorescence lifetime image shows that there's a number of, sort of lifetime changes suggesting FRED in this tissue slice. And you could then build the histogram of lifetime distribution and measure FRED. That means you could really get a local pixel-based information about protein-protein interaction in different life samples. Another set of techniques that are less commonly known, but in my opinion extremely powerful, are these correlation-based techniques. So this is like fluorescence correlation spectroscopy or image spectroscopy, correlation spectroscopy, that essentially comes in different flavors, but one of them is fluorescence correlation spectroscopy, is where you have your confocal beam parked somewhere in the cell, and let's say you're interested in measuring local concentration of your fluorophore protein or whatnot, the protein mobility and protein-protein interaction. So this is a powerful technique to do that. So you park confocal beam, and this is essentially diffraction limited spot, park anywhere on the cell, on the membrane, somewhere inside the nucleus cytoplasm, and you watch fluorophores coming in and out the confocal valve. And that creates fluorescence fluctuations. This is usually a single molecule technique that works on population, small population of molecules. In other words, if you overexpress something, you're not going to see this because it's not going to be much fluctuation going on. But small molecules, number of molecules coming in and out of your confocal volume will create these fluctuations, from which you could generate this other correlation curve. So as your other correlation decays over time, and from that, you could get this other correlation function. And so the decay time of this other correlation essentially is proportional to diffusion coefficient. So you can extract the mobility of your molecules from this function, and extrapolation to time zero will give you essentially concentration number of your fluorophores in your confocal valve. That number of fluorophores is essentially sort of inversely proportional to the amplitude of your autocorrelation function when you extrapolate to zero, which is a bit counterintuitive, but you can think of it that really, if someone leaves this room, it's going to be small disruption because people would barely notice. But it's like three of us sitting here and one left, it's going to be 30% change in signal. So the smaller the number of molecules, the greater is disturbance and then fluctuations, and therefore you could basically measure your local concentration as long as you calibrate and you know your confocal volume, which is in femtoliters. So it's a really small confocal spot. In true photon excitation, you could get even smaller and measure this fairly precisely. These are just examples of other correlation function that measured in solution for small molecules in cytoplasm in the membrane. So you could see that the decay time varies greatly, and you could sort of extract diffusion coefficients from this technique. Now, the limitation of this technique is really that it's low throughput, because you have to make your peak spot, you park the beam, and you have to spend like 20, 30 seconds to measure this autocorrelation function. Then you could go to another spot. So sort of low throughput in a way. For mapping, like globally, there are a number of other techniques, including raster image correlation spectroscopy, RICS, which basically does the same thing as this previous technique, but instead of parking the confocal beam, it actually does the same scanning as normal confocal microscope would do. So scan your beam, but just have to adjust the rate of your scanning, the horizontal scanning and vertical scanning, to sort of your expected diffusion coefficient. And as a result, this is like now beam moving left with this little circle left to right to scan. And so depending on your diffusion coefficient, as molecules diffuse, at this time point, you lose correlation. So this is going to be 100% correlation as you go down the scanning thing. And you kind of has the spatial correlation function from which you could determine your diffusion coefficient. And so this is like one example of looking at the diffusion map in the nucleus. And so you could basically appreciate that the diffusion is slower in nucleolus compared to the rest of the nucleoplasm. So you could essentially determine this diffusion heterogeneity in your organelle cytoplasm over mass. So this is really a fairly powerful technique. And finally, there's some image correlation spectroscopy that Yan Cheng will tell you more about. When you do this, basically acquire a series of images, stacks of images, and look at correlation or fluctuation of intensities for every pixel. And from that fluctuation, you could basically extract the oligomeric status of your protein. The idea being is that it's like these are protein oligomers. They'll cause larger fluctuation amplitude in your pixel or volume as opposed to monomers. And from that sort of fluctuation around the mean, you could basically determine the oligomeric status of a protein. And I'm going to dwell quickly on some of these super resolution imaging techniques. And I really classify them in two categories. They all call themselves super resolution, but I just basically consider some of them are improved resolution. They did really below diffraction limit, but within twofold. And so some of them are like structural elimination microscopy, area scan, selected plane could be also sort of improved resolution, etc. And there's really nanoscopy, which goes down to better than 50 nanometers resolution, so essentially getting close to 10-fold improvement of resolution. Those are STED, stimulated emission depletion microscopy, Palm and Storm. I just spelled them out here. But sort of the Palm and Storm are not readily compatible with LiveCell imaging because it's a long-term acquisition and sort of you need fixed sample to really reach the proper resolution and precision. STED, however, is compatible with LiveCell microscopy, and Yan Cheng will tell you a little bit more about it. This is like one of the sort of improved resolution techniques that we have in the lab is Zeiss area scan, whereby they essentially replace the whole confocal system with pinhole with this array, honeycomb array of 32 gas detectors, and you collect all the light. This is your regular point spread functions with ripples, and then computationally assign pixels and get about 1.7-fold improved resolution. And this is like just a reminder what sort of this area limit is, because the point spread function looking like that, not sharp enough, and too closely spaced fluorophores are not really resolved here. And there's a number of ways to do that. As the STED, as Yan Cheng will tell you more about it, it uses essentially point spread function engineering when they deplete the peripheral light here emitted by fluorophore and make this little nice and tight point spread function, and you get better than 50 nanometer resolution. Palm and Storm essentially doing single molecule localizations and precise fitting to get the location of individual molecules excited or activated one at a time. And then structural elimination essentially uses this structural elimination pattern when they rotate the grid and then get this high frequency moire from which they sort of get twofold improved resolution in 3 dimensions. This is just an example how it's done. And so sort of variation on a theme, Palm is photo activation localization microscopy can also be used for LiveCell imaging in different twists. So you could basically activate individual fluorophores on a cell and then track motion of this individual fluorophores before they photo bleach. For that, you get sort of essentially the trajectories and diffusion properties. So let's say this is an example of different single molecule localization trajectories, and you could distinguish by looking at mean square displacement and other ways of analyzing it, the directional movement from restricted diffusion or normal diffusion. So kind of tell locally what molecules are experiencing it, and that could tell you about interaction, transient interactions, and active transport. In the few minutes left, I'm sorry because of technical glitches, I'm just going to do a shameless plug and show some of the examples of LiveCell imaging that we used in very simplistic manner. So for instance, you could look at molecule or particle endocytosis. In this case, it's an HIV virus. By using essentially nothing else but the pH-sensitive fluorophore. Ecliptic fluorine is a version of GFP that is quenched by protonation. So it actually its pK is around 7, so it's half quenched at neutral pH. And mildly acidic pH would quench it. So if you co-label the virus with the red core fluorescent marker and have this green fluorescent marker pH sensor on the surface, then sort of entry, these are cells decorated with double labeled yellow viruses, and as they get into the compartment, you lose green signal and maintain red signal, and that could be done on single particle level. You see this sort of loss of green signal here, and this will be the movie to show that. So this is a perinuclear area where particles actually been transported to and turning red over time. So, you could actually measure the rate of endocytosis and rate of endosome acidification around your particles. You could actually get more fancy and measure the actual pH around your particle or virus. In this case, it's a tandem FRET pair of teal fluorescent protein, yellow fluorescent protein. And yellow fluorescent protein is also pH sensitive. It also quenched at acidic pH. So normally at alkaline or neutral pH, there's FRET. Once you get into acidic pH, yellow fluorescent protein is quenched, there's no longer FRET. And as a result, you see a blue shift in fluorescence between pH 7 and acidic pH here. This is done on spectral confocal microscope. So you see these two peaks, the cyan peak and yellow peak, and the relative amplitude changes as you titrate the pH around particles on the cover slip. You get your calibration curve, and from that, you could actually measure the pH on around single particles, in this case, 3 color measurements. Red is GFP or MK, red fluorescent protein trapped in the virus, and then the tandem fluorescent protein with blue and green on the surface. This is the pH sensor, and this is the content marker that will reduce the pole fusion. So this particle look initially whitish, will turn purple when you lose green signal at low pH. This is now acidic compartment here. And then it will turn blue as you lose the red content marker. And this is the movie. So that here, this one. This is like fluorescent, autofluorescent perinuclear area. Particle going to shift towards that area, turn purple. And so you can basically see this virus entering into the acidic compartments and fuse in real time. And you could basically reconstitute this, measure the pH around particle and time of fusion. And then different labeling strategies. One could essentially tweak the labeling strategies and do label integrase as a core marker. And then we've sort of developed this cyclophilin-based red-based marker that sort of binds to capsid tightly and label the capsid. And one could sort of follow the cytoplasmic transport of this intact course, and then the sort of vacuum in the nuclear membrane and loss of capsid based on loss of cyclophilin-based red. This is a three-dimensional trajectory of the particle, vacuum at the nucleus and then entering the nucleoplasm. So it could do a number of things, and double labeling essentially gives you some sort of way to assert functionality of your complexes. Let's say there's fusion going on, whether this is the content or it's encoding, sort of gives you some sort of a sense of more of a productive entry as opposed to sort of following the particles that don't do anything reasonable. So and maybe spend a couple of minutes showing another sort of way of doing it, again FRET-based. So this is a DPR-based bifunctional marker. It has YFP in green and cherry in red, linked by a short linker that contains 2 protease cleavage sites. So upon maturation, these 2 fluorophores are separated. And so therefore, one could achieve nearly 100% of co-labeling efficiency. And the sort of released fluorophore is supposed to be released upon viral fusion somewhere, which could be emulated by mild lysis of fluorophores by saponin. You could see that some particles turn green, they release this content, the surrogate for fusion, and some didn't. And so why is that? And the answer is very simple, because that cleavage is not 100% efficient, so some remain uncleaved, and that leads to FRET between YFP and cherry. And so the ones that are cleaved, they lose FRET. And essentially, here's FRET measurements in magenta, and you can see that really good anti-correlation between FRET signal and lysis. So the guys that exhibit FRET, they don't lyse upon saponin exposure, and the ones that don't actually lyse fairly efficiently. So that could be used also to really assess in real time the maturation status of newly or nascent viral particles based on this recorder. And also 100% colocalization of the TOR markers, of the fusion marker, and cherry and core marker, is really sort of an important step to try to sort of implement correlated fluorescence EM imaging that Francis is going to talk about. So you could see that basically this guy's sort of turning green, releasing in cherry content, is really a marker for fusion. You could find these particles on EM grid and then zoom on and then do cryo-electron tomography and hopefully find early post-fusion intermediates and some sort of capsid, which also Francis will introduce in more details. And so those are sort of additional links that you could sort of check out for tips for LiveCell confocal microscopy, some review articles, and some review of equipment.
So today, thanks for introduction for all the technique. And today I'm going to talk about more in-depth super resolution and also correlation spectroscopy. So today, I think my idea is just show you amazing images and then pull you back to reality by telling you the practical aspects of each technique. But I would say I'm not the expert for each technique, but I will try to go through the big points or from my hands-on experience to tell you what you need to be aware of. So those two are the great images, videos from light sheet microscopy. So you can see the live cell imaging of T cell and the target cell and see the immune synapse. And also you can see like different cellular compartments in live cell, how they interact with each other by using this amazing technique. Okay, starting from fluorescence. Usually you will see the absorption and emission from the spec of the company. And so back to the original principle, we can start with the Jablonski diagram, which has multiple excited states and electronic states and vibrational states. And you can have transition from ground state to multiple excited states, and then shortly after the relaxation, the fluorescence will happen from excited state to ground state. So since there's multiple transition from different states, you will combine together, and then you will have this absorption and emission peak. At the same time, the excited states will go, some portion of the electron will go to triplet states, which is another electronic states. The lifetime of triplet state is around microsecond. So this I will mention a little bit in FCS slides. So just take home message is fluorescence time is around nanosecond, and phosphorescence, the triplet states, is around microsecond. And also, if you go through multiple cycle for your fluorophore, a little portion of the electron will have the photo bleaching, which is irreversible. So that means your fluorophore is destroyed by, for example, the photo-induced damage. You can change the chromophore structure, destroy the pi-pi interaction, something like that. And oftentimes it's from the reactive oxygen species, ROS, which can interact with your chromophore and then destroy the structure. And then you will not see the fluorophore anymore. So this will create the idea of a photon budget. So your one fluorophore, oftentimes the photon budget is 1 million photons. So this brings us a way to think of how to design our experiment. Because if you think of the illumination intensity, starting from when we're sitting here, the room lights, it's 1 minute watt per centimeter square, to sunlight and wide field, and then confocal. So wide field is around 1 to 10 watt per centimeter square. It's a little bit higher than how you feel stands under the sun. but it's not that strong. But if you do like confocal imaging yourself, it probably will hurt a little bit. So in this case, we need to think of the illumination condition for different systems. And so of course, if you apply stronger intensity for stronger illumination, you will have a better signal because you beat the fluorophore more. So you can create, you can collect more fluorophore from your system. So in this case, confocal generally it's like 1 to 10 kilo, like 1,000 photons per second. And for wide field, it's like 1 to 10 photon per second. So the signal to noise ratio is something you can think of. And so at the same time, since the signal to noise ratio, it depends on the photons, and the photon budget for each fluorophore is like definite number. So that means you have limited time for you to do the observation. So which means, for example, for confocal intensity, if you achieve like 1 to 10 kilo, like 1,000 photons per second, your observation time is around 10-ish seconds for one fluorophore. So you have like this amount of time, and also you need to think about the photo damage you apply on your system. So this is the general idea when you do LiveCell imaging, how you want to collect your sample and how long you want to do the imaging. So just briefly go through the fluorophore I will mention today. The first part is fluorescence protein, and the second one, organic dyes. So fluorescent protein is around 27 kilodalton, and organic dyes, for example, silicon rhodamine, which I will mention a lot today, is like this structure. So it's like much smaller than fluorescent protein. And when we compare the fluorescent protein and organic dyes usually have better photon budgets than fluorescent protein. So that means for your system, if you want to have better signal to noise ratio, or you want to image longer, organic dye will be better. However, fluorescent protein, because you can just put the DNA sequence to your protein, so it's easier to do the labeling to LiveCell. However, for organic dye, there still have some way you can do the reaction and then tag the fluorophore to your target protein. For example, halo tag, you can have also like fusion protein with a halo tag protein, and then do the reaction for the fluorophore to the halo tag. So it's a similar idea to fusion protein as fluorescent protein. So just want to introduce one special kind of fluorescent protein called photo-switchable fluorescent protein. So this fluorescent protein, so for general fluorescence, after the excitation, you will see the emission, the fluorescence. But this special type of fluorescent protein, it has engineered dark states, which will fluorescence fluorophore will go to the dark state. So this dark state is engineered by the mutation of fluorescent protein, and then it have a very stable dark state. So when you do the imaging, you will see the quickly bleach to dark state, so you will not see the fluorophore again. However, scientists can use a second laser to make the fluorophore bright again. So it's a special protein that you can photo controls by using second laser to make your fluorophore to be bright again. So it can manipulate or modulate your fluorophore by this second laser. So this type of protein is widely used, especially for STAT and also some other kinds of modulation technique, which I will also mention that in the following slides. So So I like to talk about the practical aspects of imaging. So when you have the sample on cover slip and you want to do the fluorescence imaging, you have excitation filter to make sure you excite the right wavelength. Go through the dichroic mirror to reflect the excitation, and then focus by using the objective. And then you will have the fluorescence go through the dichroic mirror and then use the right emission filter to collect the fluorescence. So if we look at the focus, because the objective will focus your light at a small focal volume, and then you will only see this region. So generally, if you use like one photon for exciting the ground state to excited state, you will see the fluorescence. However, because the when you focus your light, the out of focus light still can induce the fluorescence. So that's something called out of focus emission, which can be your background. So scientists use another way to overcome this limitation by using two photon absorption. So two photon means you have two photons encounter the fluorophore at the same time within the quantum events. So that means your two poles need to be very, very quick to interact with the fluorophore like in the same time. So that means your photon can be like half of the energy of this transition. So the wavelength can be like twice of the wavelength. So it's a longer wavelength excitation. And also because the chance to hit the fluorophore at the same time, it's very limited. So the sectioning is much better than one photon excitation. So since the great sectioning from two photon, and also the longer wavelength will have less scattering for like tissue imaging. So it can go to deeper penetration. So it's widely used in live cell imaging or tissue imaging or like animal imaging in this case. So another thing of imaging is that when you want to see a point, you also need to consider the point spread function of your optical system, because you can think of the light as kind of like a wave. So you will have the interference. So in this case, the point spread function showing here, it's not unlimited resolution. So in this case, if you want to see like this line A, if these 2 points are far enough, even convolute with the point spread function, they are still far enough to resolve that. But if you are looking at B, these 2 points are too close within the limitation of the point spread function. You cannot resolve by looking at the image. So this is the idea of diffraction limit, which Greg also mentioned a little bit in his presentation. So generally, the diffraction limit, the definition is like wavelength over 2 times numerical aperture, or you can just simply think of half of your wavelength. So in this case, since our current optical system, we cannot image better than this diffraction limit. However, for our like a sample, if we want to image like a protein structure on virus, it's like within diffraction limit. So it's really hard to use conventional microscopy to image that. So that's why we want to do super resolution or nanoscopy. So just give you one example. This is what we did in STAT. So we have a GFP VPR, which in the core of the HIV, and then we stain the envelope protein, which on the surface of the HIV. By looking at the confocal image, they just simply merge together. We cannot see any structure. However, if we use that, you can see 2 clusters here. So this is a way to see beyond the diffraction limit and study the protein clustering, and maybe it can indicate how your like virus maturation or some other kind of phenotype. So the first technique I like to introduce is called stimulated emission depletion microscopy, also called STED. So back to the theory, after the excitation, like the absorption and emission, it's like give you the conventional resolution of fluorescence microscopy. So you will always have the convolution of your point spread function. So for example, if you want to see the structure, they just like simply merge together. But if you use stimulated emission depletion, so this is a second laser which can force your excited states back to ground states. So it can deplete your fluorescence by using this second laser. So in this case, scientists engineered the point spread function by using this donut shape of STED beam. So it will only deplete the fluorescence around this excitation beam. So after these two overlap, you can get the point spread function to be much smaller. So in this case, you can have like much clearer image compared to the original image. So by looking at this, currently the market has like some STED microscope which is available. First one, Leica, they have a resolution XY like less than 50 nanometer. Z is like 120, and a barrier Steadicon, they have like 75 to 90 nanometer for XYZ. So if you guys are interested in, you can contact them, even though I do not have the share from them. Yeah. So for practical aspects of how you design your STED imaging, so for for example, this Alexa 647 excitation emission, and then you want to see which STED beam is good for your STED imaging, you need to make sure that your STED beam, so this depletion beam, have the significant emission overlap, significant overlap to your emission spectrum. So for example, here, the depletion beam 775 has a little bit overlap to the tail of your emission spectrum. So this is the basic idea of how you choose your fluorophore and how you choose your STED beam. So this is the example for Alexa 647 by using 647 excitation and 775 STED beam. And for the same setup, you can also use Neptune 2, the fluorescent protein, or the silicon rhodamine for your system. So, okay, so if we want to do true color, how we do that? So here I'm showing you two fluorophore. So the dashed line is the absorbance. So two absorbance here, and two emission is the solid line. So the dye one star 440, you can excite with the 458 and emission like collecting here, and then dye two excite 514 and collect your fluorescence here. And since their fluorescence spectrum have the overlap at the tail region, so they can use the same STED beam for depletion. So this is another way you can choose your chromophore and to do the STED imaging. So in this case, this website is very useful. If you're interested in, they list like a multiple chromophore with like different STED beam. So you can, based on their information, to choose which chromophore you want to use and which STED beam you want to do for your STED imaging. So one practical aspect is a cover slip. And I think this is very important, and sometimes I will just like forget this part. So cover slip, you just need to make sure your cover slip is right for your objective. So most objective, it's for 0.17 millimeter thickness. And I know it's a little bit confusing because the number is 1.5, it's not 0.17, but just make sure that you have the right manufacturer thickness for your cover slip. And if you are worried about the quality, you can use even higher performance, 1.5H, have a better quality control like a cover slip. And the second part, if you want to do LiveCell stat imaging, just make sure your system is good for LiveCell to like keep alive and happy. So you can so using R as the reactive oxygen species scavenging buffer to protect your fluorophore, and using far red depletion excitation to make your cell happy, minimize the cell stress. So this paper is very useful because they mention different aspects on how to keep your cell or keep your live cell happy for the live stat imaging. And of course, the live cell imaging, you need to fuse your fluorescent protein or using organic dyes with SnapTag or HaloTag. So here's are the imaging of your. So this is the tubulin with the silicon rhodamine, and then you can see the they are moving, and then the resolution is really good. So this video is from a barrier image with Steadicam, and the second example is from this PNAS paper. They are looking at the morphology of PSD95 in live mouse. So this protein is at post synapse, so it can indicate some functional aspects of your synapse function. And they are doing the surgery on the mouse brain, and then inject the LiveAct EYP to see the synapse structure, and also using the HaloTag for silicon rhodamine to target the protein. So in this case, they can observe the live imaging on the mouse brain and see how the morphology really looks like. So by looking at the confocal, they are just like a cloud. But using the stat, they can really see the morphology in live mouse. So this is a great example by using stat on live imaging. So just want to show you some useful papers and for stat. So of course, the inventor for stat technique is Stefan Hell, and the second guy is a PhD from his lab, and he actually have his own lab. So following these two labs, you can see what's the most advanced technique and what's the most advanced research. And the first paper is showing you the live cell stat microscopy, how they assessing the fall damage, and then how to minimize the cell damage. And the second and third paper is a conceptual idea of stat. So yeah, if you guys are interested in, you can take a look, or we can discuss after this session. And then the Leica's website, they have a very practical points that you can start from choosing your dye, choosing your laser, and how to prep your sample. So of course, different technique will have the disadvantage of the technique. So the biggest disadvantage of stat is it has a very strong power to deplete your fluorescence. So in this case, the stat beam for depletion is megawatt per centimeter square. If we go back to the a little information I provided before, wide field, confocal, and then stat is kind of 1,000-fold increase. So in this case, you really need to worry about your how severe you will photo damage your sample, especially for LiveCell imaging. So actually, so the biggest caveats is the photo damage and photo bleaching for your the LiveCell imaging. So actually, they also developed another a little bit modified version of STAT by using this photo-switchable fluorescent protein because it has engineered dark states, so they can use much less power for deplete the fluorophore. So this is called resolved, and yeah, they can get similar resolution by using this technique. But at the same time, resolve because for this photo-switchable fluorescent protein, it needs to use 4 or 5 lasers, so which is blue laser can also create some damage to your sample. So this is also another caveat of this technique. So the second part I want to talk about is the correlation spectroscopy, or called FCS. So FCS, when you look at the, your setup, you have your focal plane in your sample, and then you study the fluctuation of your focal point. So this fluctuation can contain lots of information. For example, the diffusing of your molecule, the blinking from the photophysics of your molecule. So by analyzing the fluctuation, you can get lots of information. So for example, if you focus in the cell, you can see the population in this focus by using FCS. So to guide you through how to do the calculation, by looking at the focal volume of the this focal point, you will have this a fluorescence time trace, and from fluorescence time trace, you can get the average intensity. And if you looking at the fluorescence at each different time points, you can have the fluorescence at this time points, and then by calculating the difference between fluorescence at T and the average, you can get the fluctuation value at this time point. And when you do autocorrelation, you can get this autocorrelation curve, and then have this autocorrelation function. So by looking at this, you can have different region. You can interpret it from this data. So from a microsecond region, because the triplet state is around microsecond, so you can get the information of the photophysics of your dye at this microsecond region. And from for this region, it's oftentimes the diffusion time. So in this case, I'm using the organic dye in the solution. So the diffusion time is around 20 to 30 microseconds. So you can see the decay, it's pretty fast. But if you do this in the live cell, for example, cytosolic fluorescent protein, the diffusion time is around 1 millisecond. So you will see the shift of the decay around here. And if it's even on the membrane, the diffusion is even slower. So you can see the shift of your diffusion. So by looking at the autocorrelation curve, the information you can get are the amplitude and the time, the decay time, and also the slope. Is it steep or is it shallow? So these 3 points can give you the information how your system behaves in your focal volume. So also the amplitude, just like Greg mentioned, the fluctuation, the amplitude is proportional to 1/n, which means if this room have 200 people, I cannot see the the leaving of the person. But if this room only have 3 people, I can obviously see the fluctuation of your of the population. So FCS, it's kind of a semi-single molecule technique, which like to see the dilute molecules in your focal volume in order to see the fluctuation. So for example, if in our lab we did the FCS in the nucleus and see how the GFP VPR behave in this focal volume. So by using the monomer GFP and tetramer GFP, we can see the GFP VPR is somewhere in between and seems to have two populations. So have one population is more or less similar to monomer, and the second population is more it's more like an aggregation form of the protein. So this is a way you can study the a population in your live cell and how they aggregate and how they change their diffusion time by using FCS. And since FCS, the diffusion coefficient is proportional to the qubit of the molecule weight, so it's not that sensitive when you're looking at protein-protein interaction, because if you think of two proteins come together, the molecular weight just becomes twice, and the diffusion coefficient is still very similar. So in this case, people are using cross-correlation by using two colors. So for example, molecule 1 with green, molecule 2 with red, if they diffuse together, if they have interaction, the cross-correlation will increase the signal for your correlation curve. So in this case, we have some control experiment. Fusion protein, of course, they bind together. So you will see the amplitude of the cross correlation. And if they are not interacting, it's similar to nothing for cross correlation. So this is another way you can study interaction in live cell. And the practical aspects of correlation, FCS and FCCS, you need to think of how to determine the diffusion coefficient in your focal volume. And since the FCS curve will only give you the diffusion time from fitting the correlation curve, so in this case, you need to find out, if you want to find out the diffusion coefficient, you need to know the geometry of your focal volume. So in this case, every time when I do FCS, I need to use a standard dye with a known diffusion coefficient to determine the focal volume at that day. So before I start the experiment, I will always use the standard dye to do this calibration. So if there's a very useful sheet you can find online from PicoQuant. They list out lots of organic dyes for you to do the calibration. So you had from this sheet, you know the diffusion coefficient for each different dye, and then you can use that for find out what's the geometry of your focal volume. And the other aspects of the FCS, FCCS, the first part, you need to make sure your pinhole for two colors are the same, because if they have a little bit shift, your amplitude will have some offset, so it can give you a wrong number. And also, if your protein diffuse slowly in the focal volume, you may see the focal bleaching of your time trace, or even the self movement of your cell membrane. So just keep in mind when you do FCS or FCCS, make sure you see both correlation curve and time trace to make sure what you have is the correct information. And actually, for this slow decay, there's some papers say, okay, they can use some correction to do this. But yeah, we can talk about that later if you're interested in how to correct the focal bleaching. So there are lots of paper and software for FCS, FCCS. I especially like these two papers. So the first paper is about introduction for FCS, and the second paper is the practical guideline for the FCCS. if you're interested in, yeah, you can also take a look and see how to set up the system, which points you need to be careful. And when I do the correlation in our lab, I'm using Zeiss FCS module and then do the fitting by Origin Lab or QuickFit. So yeah, so this is the way I do for FCS. And since FCS, the limitation is only looking at one focal point for like 10 to 20 seconds. So it's really hard to study the whole information from the entire cell. So that's why people are developing image-based correlation. So today I'm going to talk about number of brightness more specifically. So if we want to move from a single point correlation to image correlation, so the reason why I want to talk about number of brightness because it's compatible to confocal microscope, which is I believe most people have used the confocal microscope in core facility or in your lab. So it's really easy to apply and then use this technique for measurement. So the idea of number of brightness is still like a scan through your image and then do several frames and then use those information to for number of brightness calculation. So the number of brightness calculation, the two information you need to have is average intensity for this like a single pixel at different time, and also calculate the fluctuation, which is the variance of your this pixel. So the idea of a number of brightness is that if you have 6 monomer, the average is like 6, and if you have 3 dimer, the average is still 6. However, the fluctuation for this two, because dimer, when you leave out the focal volume, you will see more obvious fluctuation. So by looking at the variance over average, you will see the difference between dimer and monomer. So this is the basic idea of number of brightness. So yeah, so you can see here the fluctuation for dimer is more than the monomer. So the calculation will be variance over average and -1. So why there's a -1? Because if you have an immobilized molecule, the fluctuation is actually from the fluctuation of your detector. So that's like a Poisson distribution. So your variance will equal to your average. So in this case, since they are equal, the epsilon will be 0 for immobilized or for the thing that's not moving. But for mobile fluorescence, you can see since the monomer, dimer, trimer, the fluctuation will increase. So you can have a greater and greater epsilon value from using this formula. So this way, we can calculate the epsilon from your time stack of your images and then find out the brightness of your sample. So for example, this is what we did in the lab. So for monomer GFP and 2x dimer GFP, the epsilon value, you can see that increase here. And they are not depends on the average intensity. So some cells, they are dim, some cells, they are very bright, but the epsilon value, they are similar. For this 3 and this 2, they are different intensity, but their brightness is similar. So this is the way to do the calculation and find out the brightness of your system. And actually, there's another derivation. They can do 2-color imaging and see the interaction, just similar to cross correlation by using similar formula, but just 2-color. So how to set up for your confocal setting is that because you are scanning through your image point by point for your confocal image, so just keep in mind that pixel dwell time needs to be short enough, like much shorter than your diffusing time. So each point when you acquire the image, it's not the average of the motion. So you can have the snapshot of each population. And for frame time, because after you scan through the whole image and then become like the second frame, you want to have the population already changed. So in this case, the frame time needs to be much greater than the diffusing time. So you can see the populational change and then observe the fluctuation from your single pixel. So this is the idea how you design your number of brightness. Just make sure your dwell time is like microsecond and frame time is like second. So your diffusing time is in between. So some example, they are looking at this GR, the protein. So this protein oligomerization states can indicate the cell like signaling. So they use some control experiment to see what the monomer looks like, the brightness looks like, and with some treatment, they can see the like dimerization. And actually for the dimerization, they can even see more dimer from the dimer, so kind of like tetramer or higher order. So number of brightness, it's a useful technique, not only give you the oligomerization, you can also observe the heterogeneity of your sample. So of course, there's limitation. So for number of brightness, since you need to acquire like one frame around 1 second, and you need to have enough signal to calculate the average and the fluctuation, the variance. So in this case, typically we do 20 to 50 frames to create one number of brightness image. So that means your dimerization or oligomerization needs to be quite stable, and then will stay maybe like 1 minute or longer in order to observe that in your number of brightness. And the other thing is monomer to dimer is not always twofold increase, because some fluorescent protein, they might have dark states or they might have like folding issue. So for example, EGFP, one monomer dimer 1.8, and cherry is around like 1.5. So this is the research from this lab. So yeah, so for number of brightness, there's some useful information. So this is the first paper is actually the original paper for number of brightness. So they developed number of brightness by using confocal microscopy. And the second paper is the cross number of brightness. So by using two color to see the interaction between these two protein. And of course, this lab from Rincon Graton lab, they developed the software called SimFCS, and you can go to their website and download the software to see to use their software to do the number of brightness. So oh, okay, I will quickly wrap up. So I just want to introduce how we did in our lab. So this is another use of photo-switchable fluorescent protein in LiveCell imaging. So oftentimes when we do the imaging, we will have the autofluorescence background. For example, you can see like a uniform autofluorescence from metabolites like NADH, FAD2, or the green or the red channel. You can see the punctate from the cells. So in order to remove the autofluorescence, we are using the photo-switchable fluorescent protein. Because photo-switchable fluorescent protein, we can use second laser to make the fluorophore bright again. And then it can be like reversibly like bright, dark, bright, dark. So we are manipulating the fluorophore by using these two lasers. So in this case, image 1, since it's before the 405 activation, so if we target or specify the photo-switchable fluorescent protein with to the mitochondria, before the activation, there's no mitochondrial signal, but after the activation, you can see clearly the activation of your mitochondria. So in this case, we can do the calculation from this two image, just simply doing subtraction. And since the background, they are not photoactivated by the 4 or 5, we can just simply remove the background by subtraction. So we apply this to LiveCell's single virus tracking. So in the peripheral, some cell, the peripheral, like a perinucleus region, it's have lots of autofluorescence. So you don't know where's your virus. But by using this modulation scheme, simply doing the subtraction, you can clean up this region pretty good, and then find out where's your virus. And then we can apply the LiveCell imaging tracking on this virus by using this demodulation scheme. And then we can get the trajectory from this. Yeah, so this is another way to use for photo-switchable fluorescent protein. So if you guys are interested in photo-switchable fluorescent protein, there are also some useful papers. So the first paper are actually the original paper for developing this type of photo-switchable fluorescent protein. And the second paper is the mutation that creates several different types. And the third and the fourth one is using this photo-switchable fluorescent protein for stat imaging. So yeah, if you guys are interested in, you can also take a look at those papers. Yeah.
So just to go through this session, I will be presenting a small introduction, brief overview of the HIV lifecycle labeling techniques and how we generate fluorescent viruses and general parameters, which have already been covered by Yen-Cheng and Greg on considerations for LiveCell imaging, and then we go straight into the particle tracking software, right? So what we study is this small beast, which is about 10 kb in 10,000 base pairs in length, and it codes for about 15 proteins. And many of these proteins are, there are structural proteins, enzymes, and there's the envelope protein, and there are a bunch of accessory proteins. The virus, all these come together to help the virus replicate in cells. So in a mature virus looks for something like this, where you have the envelope on the outside, you have matrix on the inside, then you have capsid, and inside the capsid is your viral replication complex. So together with the viral replication complex and the capsid is called the viral core. Okay, so these terminologies can vary from people to people, and but it definitely helps in when you come to decide what is it that you want to label in a virus, whether you want to label the core, something inside the capsid, or on the envelope. So you need to know how they are actually organized, right? And in addition to that, HIV infection actually progresses through very different, distinct steps. You start off with binding receptors and co-receptors at the plasma membrane. Then you fuse into the cytoplasm, where you deliver the conical capsid structure. At some point, this conical capsid structure is disassembled, a process that is fully termed as uncoating. And then you have this nuclear import step, where the virus is transported inside the nucleus. During this time, your DNA is synthesized, and then your virus integrates. And once you're integrated, you behave like a cellular gene. You make use of cellular host transcription translation machinery to make RNA get out, and then you assemble at the plasma membrane. So there are really distinct steps in viral replication, and all these steps are regulated by the ability of the virus to interact with host factors. So you can basically label the host factors and visualize virus infection, or label the viral proteins and visualize how it behaves inside cells. And these interactions that allow the virus to progress through this replication are controlled in a spatial and temporal manner inside cells. Like Greg mentioned, life is actually in 4D, right? So you need to really understand where certain of these steps are taking place, and that will give you a lot of information of what proteins might be actually relevantly interacting with the virus in those locations, because proteins are organized in a spatial manner inside cells. And there are a variety of ways to label. First, you have to choose the step that you want to study. So once you know this is the step I want to study, either fusion or trafficking or encoding or transcription. So based on the step you want to study of the virus, then you use any one of these small number of examples that I put together here that can label as a reporter for fusion or code trafficking or nuclear import or even transcription and assembly, right? And out of these, there are a few, these number of fluorescent probes. I might have missed some, but these number of fluorescent probes remain attached with the viral core. So they remain inside the core, or they are bound with the capsid. So they can be visualized for long periods of time as the virus enters the cell and gets into the nucleus, right? So once you've chosen this marker, an example of that. So what I've just told you is that these are all single markers, apart from this underlined marker, which is a 2-color marker. Everything else is single marker. When you use 2 colors, one as a fusion marker and the other one as a core marker, then you can see a loss of color during fusion. So that gives you a readout for a particular step in viruses. So when you combine 2 markers, you can actually gain functional dynamics of viral steps, right? So in this case, is an example that we have developed in our lab, where you have the CPDS protein, it binds the capsid. So it's organized, we believe it's organized something like this. And then you have the integrase, which is the core marker, which is inside the capsid. And then you can infect cells and you will see the loss of DS red signal. And whatever loss we see, we interpret them as uncoding, right? So you can see gradual loss, abrupt loss. We have described that in several papers. And then this marker is also useful because you can actually look at cores inside the cytoplasm, and they will be double colored because they will have capsid and integrase signal. And if you add this drug cyclosporin A, it will competitively displace the CPDS, right? So you can actually tell whatever core that lost the second color is actually in the cytoplasm and not inside endosome, because inside endosome, both the colors will be trapped in this small endosomal vesicle that they will not be released, and they will be seen as double colors, right? So these are some of the ways to use 2 colors to define functional steps of the viruses, right? And so how you generate fluorescent protein, you first need a plasmid that has the viral backbone. It codes for the viral RNA. In this case, it's a PR9 vector, I suppose. It has all the viral proteins and the RNA genomic element, but here we use this delta envelope plasmids because they are single round infectious. They cannot make infectious virus per se. And then you can combine them with your fluorescent label, a second plasmid. A fluorescent marker can be any of these things: VPR, integrase, A3F, GAG, GFP, CPDS. You can put those plasmids along with a third plasmid that encodes for an envelope protein. Okay, so envelope is required for virus to be able to enter your target cells. So, and this type of labeling is called pseudotyping. You take these 3 plasmids, you put it inside 293 cells. They produce high titers of virus, and they start generating viruses. You collect the virus, and then you can have a fluorescent virus produced in this manner. You can put this virus on cover glass and do a lot of characterization on how the fluorescence looks like and what they can report and what they cannot. So when you take these viruses and you go to do LiveCell imaging, some of the things that you want to consider is that the fluorescent protein that you're attaching to your virus needs to be bright, sufficiently photostable, and it should not affect virus infection, because you're studying virus infection, then you don't want to really affect the virus infection with your fluorescent protein. And then you can perform validation experiments. Then you take these viruses, once you know this is a good fluorescent label, you take these viruses, infect cells, and you're ready to start your imaging experiments. But before you go to your imaging experiments, some of the things you want to consider are your excitation parameters, or the imaging parameters, as I would say. So you want to be at an optimal laser intensity that gives your signal above background, and you can detect the fluorescent viruses. So there are some tips I've just put down here, how to calibrate your microscope system to visualize viral particles efficiently for LiveCell imaging, right? And then you test for photobleaching, so you can expose your viruses to so many number of times, so many light cycles before they bleach. So what is the threshold of fluorescence that lights you can shine on them, and then you can calculate the number of frames or number of times you can image them, or number of frames. For example, if you want to do a Z-stack image with our conventional LSEM, if you do 240 frames of 13 Z stacks, and then you want to go, you can do that in 2 hours with the imaging frequency at 30 seconds, or if you do it for 20 hours, then you can image them for every 5 minutes. You can even do longer by extending your like delaying your frequency of imaging time. So once you have figured out all these conditions that you want to use for your imaging purposes, you are ready to do the imaging, and then you can basically put your viruses and is it playing? Well, something happened there. You can put the viruses there, and then it just got blocked. Sorry. Yeah. Okay, so you put the viruses on cells, and then you can actually follow them. That was a video, but hopefully it's not like. So you can put the viruses on cells and acquire frequently time-lapse images, and then you will see the virus spots actually being over here. It won't play. It's okay. Yeah, so you would see spots like this inside your cells. Something the virus punctate will appear like spots. Each spot has its own property, like it's basically a fluorescence property. It'll appear like a peak. There's a PSF. So all these properties of spots go into algorithms of spot detection. So your single particle tracking involves 2 steps. One, you detect all the spots in your image, and then you start connecting the dots to see the trajectory of your viral particle as it goes inside cells. Okay. And there are, like I mentioned before, there are several softwares to do this. And today I will give you a primer, hands-on experience on this IC image analysis software to do single particle tracking and image.
We lost you again somehow. Really? When you went out.
Okay, back in. Okay, now share. Is it better? I think. Yeah. So we will go through this. So IC image analysis software is basically was developed by a bunch of consortium of microscopists and image analysis individuals, founded in Institute of Pasteur and France Bioimaging, and they are based out of Paris. So you could have might have downloaded the software from this website, and it's pretty cool because what they believe is that they actually want to develop, because like the type of experiments we do in our lab is probably not the same that another lab would do, and so the way you analyze your data might be different. So these guys help you tailor your analysis protocols for each lab. If you write to them, they will also answer. I really like them for that particular reason, and also for the software. So if you had opened the software and browsed through it, just going through briefly, it has it looks like this, and you have the first sequence where you have, these are organizational tabs on top. Then you have the file importer exporter tab over here, and then you will go through the series of tabs that allow you to do series of function. For example, here you can crop your images in XY or in Z or in Z, or remove channels, remove frames, etc. Right? Then you go to the next tab, and it's got region of interest. This is probably a very useful place to be in, because it allows you to draw rectangles or circles around the area of your images and just observe, just do your quantification in those areas. And then you have ImageJ. There's a built-in ImageJ plugin over here. Basically, it talks to the ImageJ. So any plugin you have in ImageJ that you fancy using for your analysis, you can incorporate that directly through IC. And then you have the detection and tracking plugin. This is something that we will go through in more detail. Basically, it has a bunch of plugins that can do a bunch of functions for image analysis, and especially for single particle tracking. The most useful ones will be your spot detector and spot tracking and track manager plugins over here. Then you go to the processing functions, and you have filter boxes. There's a bunch of other plugins that can do various different functions for fluorescence image quantification, I would say. And then there is this is something very useful when you want to set up protocols for your software to do multiple series of functions over time. You can even do batch function. You create, basically you start writing protocols and then it's just pulling plugins together and drawing lines. And there is actually a video tutorial of this in the IC web page, and I would recommend that you guys watch this so that you can build your own protocols inside this software. Right, and then you go to the plugins version. So, and then if you go to this online plugin column over here, you will open these preferences. Perhaps some of you all have not yet gone here and downloaded ECCLAMP, but which we'll do at some point. So once you click on the online plugin, this tab opens up and it has a bunch of plugins over here, and new plugins are being added by the day. So you can search for something over here, they'll pop up, then you can just say install, and it will be installed into your IC software that you have. Okay, and then we will cover the ECCLAMP part here. And like I said, the coolest thing about IC is the support that they have here. You go here and it's basically a Google forum. You just go there, you search for a problem that you have or a type of quantification that you want to do on your fluorescence image, and people might have already done that. It'll pop up, and then you can look up what they have said and use it. Or if it's something very new, you can just write, this is what I want to do, and this is I don't understand how to do it. These guys actually get back to you in a couple of days, and they've actually built on some protocols for me at some point, you know, because they help microscopists to ease their image analysis by doing this type of back to front forum. I really like this version of this function of IC. So let's open IC image analysis software for those who have downloaded it, and we'll just go through the data sets. So today, the workshop, what we're going to do is data set, single particle, single virus uncoating the cytoplasm. So these are viruses that I showed you before with the labeled with two colors. One is red, spadius red. The other one is green. And we will try to track these particles inside these cells and evolve a trajectory and try to gain some fluorescence information from these trajectories. Okay. So moving forward, if you have opened your IC plugin, so you go to your detection and tracking. I just want to go through in detail about this spot detector before we start. So if you go into the detection and tracking, you'll find this spot detector plugin. You click on it, it opens like this. And once you open the spot detector, you can also open the spot tracking plugin. So you see there is select detection results here. So so basically the spot tracking uses these two functions. So you detect spots, and detection spot, you want to say what is your input file. So input will be always, it's set to always the current sequence input detection. So if you have an image open on the software, that is going to be your current input sequence input detection. So you might have situations where you have multiple images open on your software, right? And only the one that you click last will be the current image that you're going to put input here. So that's what you got to remember about this input section, right? Then you go to pre-processing. So if you'll have the spot detector open, just click on this channel drop-down menu. So you will see a variety of channels. Do you see a variety of channels down there? Yeah. So it basically tells you channel 0. Then they call their first channel as channel 0, and the second channel will be channel 1, channel 2, and channel 3. Sorry. Okay, so we're going back here, right? We start from here. So you got that, right? So the input, the current sequence is basically the image that you click on inside your workstation. And then you have your pre-processing. That is the second step down. So it's, as you can see, it's a very intuitive software. You know, I learned it by myself in a way, because you just go, there are only a few selections over here, and there's very little that you can do in this side. And then you just have to go through this workflow, which is given on the left, okay? And so the pre-processing will tell you, okay, this is the channel selector pre-processor. So if you have an image with multiple colors, like the ones that we have, right? It'll give you the number of channels that are here. Your first channel is always your channel 0. The second one is channel 1, channel 2, and so on. So you need to, from your image, you will already have your metadata loaded onto the IC software, which will tell you what was the first channel. It might be acquired for GFP, and what is the second channel. It might be acquired for DS Red, and third channel could be a PMT, and the fourth one could be a DAPI. Okay, so that's what you want to do here. So you select the channel which represents your virus color. Okay, the fluorescence of your virus. In this case, in our case, it's going to be channel 0, right? And then you go to the next step on the settings, which is a detector setting. So this is where you detect your objects. Okay, so this spot detector uses a wavelet function. It's basically your spots appear as the PSF of your spots appear like a Mexican hat. So you have a peak and you have a tail. So this spot detector makes use of all these information to actually calculate where is your spot. And it has these different values. You will see here that it asks you, what is it you want to detect? You want to detect bright spots over dark background or dark spots over bright background? What is it that you would expect to detect in your fluorescent image? Like, you want to detect bright spots over dark background, right? So, but if you go on an EM image, it will actually be the opposite, right? Because you're going to have electron-dense black structures. So you want to detect dark spots over there. So there you would use the other one. Then you have these functions over here. These functions are useful for a 3D image when you do not sample more frequently. So there is an optimal sampling in 3D to catch your object in more than 2 slices. Okay, so when you can catch your object in more than 2 slices, then the plugin will easily identify it. But when you do not do that because you want to avoid photo bleaching and you want to go faster, etc., so you can skip a few of these Z stacks and you will still see a particle 1 or 2 of them, right? And then if you use one of the forced use of 2D wavelets, etc., these plugins, if you click here, it will actually catch your object, 3D object, as if it was a 2D object. Okay, so that way you can detect that. And then you have these scales. So these scales, as you can read there, it's called scale 1, 2, and 3. Then you can add more scales. For example, you can just click on add scale. Somebody can click on add scale. So next one is what? 13 pixels, right? Yeah, so you can increase the size of your object. So basically, if you have a nuclei which is very large, then you want to go at a much larger scale and then detect that, right? So what the scales mean is that if the scale gives you the optimal, so if it's a 1 pixel, then you're going to get a lot of noise because it's going to look for 1 bright pixel and then the tails of the Mexican hat around this 1 bright pixel, okay? When you look for 3 pixels, which is a more reasonable number for a 512 image. So you have 3 bright pixels in between. Look for objects with 3 bright pixels in between and the hat formation following that in the circle, right? So and then you can do the same thing for 7, 15, depending on your zoom and the pixel sizes, etc. Okay, so this is very important. I think I gave you guys a protocol which works very well. So the protocol that I've given you will say use this at 80% and that as 100%. So when you combine these scales, it will help separate 2 objects that are too close to each other. Okay, so that way you don't look at 2 objects as one. All right. So these are nice functions on this. And then what you do is then the most important thing after detector, I'm just jumping straight into the output. Okay. So before you start your spot detection, you want to go to this output. So the first version of the software, when we started using it, it had this automatically enable automatic, enable specific. So what they were doing was basically, as good microscope, good image analysis should do, they were saving every step they were actually performing on the spot detection. But what that did was it'll use up a lot of your RAM and it'll kill your computer, right? So they actually left that choice unclicked. So now they will say, if you want to save your spots, then you want to click these things later on. But all we want to click, because our computers are, not strong, like not very high computing computers. So all we want to click is this export to swimming pool. So swimming pool is basically basically what, like the word says, is a swimming pool. Everybody jumps into it, right? So whatever detections you put, you throw it into the pool. So once you throw it into this pool, then you can access these detections from any of the other plugins that IC has. For example, the track manager plugin, okay, where you want to do the tracking. And after that, you go to the last one is the display. So here you can, based on what you like, whether you want to see your detections or not, you can choose or not choose. I normally leave this open, okay? And now you can open the image that I sent you. So did I send you 2 images last time? Did you guys have data set, single particle tracking, and did you have two folders? So two folders, huh? Go to the uncoding folder, okay? So we'll just track this one. So you start the software by just clicking on it. And it'll load up. Okay, so it's loaded. Now you can open your file, go to the open folder in the images sequence tab, and then you can find your folder wherever, find your folder, data set. Okay. All right, so you will see an image like this. Do you see an image like this? Yeah? Okay, to the right of the IC software, you see this series of columns, right? These series of columns gives you the channels, the original channels that were acquired on the image. This is, for example, the green here. You can actually change the color by clicking on this this location. You can actually change the color of your channel, right? So it's basically pseudo coloring. You can give it white, red, blue, whatever you want. So now we have in this image we have 4 channels. Is that correct? Everybody gets 4 channels in the image. You get 4 channels on the image. Do you see 4 channels? These 4: green, red, white, and blue. Yeah. Okay. So now you can actually turn off the PMT channel because it's probably not useful. And like I said, the channel that we are going to track in these are the green channel, the first channel that you're looking at. Okay. So in this case, you see in the channel box, you see these threshold lines. Do you see the line blue and red? Do you guys see that? Right. So these lines actually help you to control the grayscale value of your image. Okay. So you basically you increase the dark. I mean, this is just for visual appreciation, right? So you increase the dark and bright. You can make it bright and see a lot of background. You can adjust it to the way you want to see it. You know, increase the dark. And you can. Did you guys notice that you can make your image larger by just scrolling up and down on the image? Yeah, so that helps you to see where your spots are. So you see something like that? Yeah. So now if you just play this, so this is a scroller that you can pull forward in time. Okay, so when you want to do that, you want to remove these auto bounds. Do you see this auto bound on top of the imaging threshold here? You want to uncheck that. Okay, so you want to probably uncheck this label over here. So because when you uncheck that, it will not change the intensity of your image as you scroll down. Did you guys manage to uncheck that? Yeah. So you're seeing this now. So you can move the small dots that you see in green and red are viral particles, and then you see a nuclei which is in blue, and then there's a scale bar below that. You can remove your scale bars if you want to. Just go to layer, and you can see scale bar on the layer. Do you see the layer on the right-hand corner? Sequence, raw, layer, history. Yeah. If you want to remove your layer, you can remove the layer. If you want to remove your image, you can remove your image, but that's not what we want to do, right? You want to keep the image on. Okay. So now you just define a region, so you see a few spots moving, right? When you play the video, you see a few spots moving around, correct? Right? So now you want to define a region that you want to track. I want to track only a few spots out of these, okay? So to define a region, you would go into the region of interest tab, okay? Choose whichever one you like. You can take a rectangle and draw a rectangle. Everybody there? Yeah? Okay. So once you draw a rectangle, now you can, so have you adjusted your viewing, like how you visualize the viruses? Okay, so you want to have something like this. You want to see it by your eye, where the viral particles are, right? And now you want to move on time, you want to move this cursor up and down to see if your region of interest covers the whole track of the virus. Any virus you take. So for example, if I want to track these two, I just do that. So I want to make sure the virus is there inside this rectangle till the end of the movie. Did you see what I did just now? Yeah? Got it? Did you guys get the virus in whichever virus you want to track? There are a few of them there. You draw a circle where you can see them throughout the movie, right? So now you go to detection and tracking. So what we want to do is we want to do spot tracking. So you can click on the spot tracking plugin. And for spot tracking, like I mentioned before, you need to detect your spots. So you run the spot detector plugin, click on the spot detector plugin, it opens it up, correct? Are you guys there? Are you guys there? Yeah? Okay. So now we will go through the steps. Pre-processing is 0. Okay. Detector, we want detect bright spots over dark background. Like what we do with the scales, you use 2 scales, scale 1 at 100% and scale 2 at 80. And then you go to output, make sure you have enabled export to swimming pool. Okay. Now you say, no, I'm just waiting for these guys. Too fast, I went too fast. Okay, so we were in the spot detector, pre-processing channel 0, correct? Then detector is detect bright spots over dark background, correct? Yeah. Then you put your scales as scale 100% and scale 2 80%, got it? Then you go to output after that. And in output, you make sure you select export to swimming pool. Okay, so we are here. Yeah, everybody? So now we just hit start detection. And did you detect all the spots? Yeah? You got the spots detected. Now you can actually follow the spots, right? So just play your movie and see if the detections are actually tracking the spots. Can you follow the spots? Yeah? Do you see? It's done a pretty good job detecting all the objects in the whole time frame, correct? Okay. So now what you do is, now you see in your spot tracking plugin, which is this one. Are you guys looking at the spot tracking plugin right now? Okay, in that it says detection set from T0 to T219, and it gives you a number of detections there, right? So that's what swimming pool has done. So what swimming pool has done is it has put it into this swimming pool, and that is accessible by the spot tracking plugin over here. Okay, this is what you see over here. All the detection that is here is here. Okay. So now you say estimate parameters. In the spot tracking plugin, you can say estimate parameters. So when you click on estimate parameters, you open a new small window over here. Do you guys get that? Yeah? Okay, so what does it say? It actually says, what type of target motion are you looking for? Are you looking for a mainly diffusive? Like, are you expecting a particle to be diffusive? Or are you expecting it to be directed motion? So I don't know. So when you don't know, you just say it's both diffusive and directed. No? So it'll make sure it uses all the calculations to do the tracking. Okay, and then you say run parameter estimation procedure. It's done, right? So now you say run tracking. And you will get a track. You'll get multiple tracks. Okay, so it opens up a track manager plugin. So this track manager allows you to do everything that your tracks have. Okay, so it allows you to join 2 tracks or analyze your fluorescence in your track, Are you guys already there with the track manager? Yeah, everybody? Yeah? You got it? Yeah? Everybody's in this step? I don't know where they're. Right? So so some of y'all might have already come to this stage, and some of y'all might have problems with the software, or you might not be in the stage. Okay? So if you just gather with the people who already come to the stage, we can move forward on this step. Okay? So what you see here is basically, you see a list of tracks. I don't need this. So you see a list of tracks shown over here. This is the track manager. On the left, it shows, because sometimes it does not show you these tracks, so you have to go to display and say display track panel. Do you see that function that is there? I went to display on top, and if you say display track panel, it will show you your tracks over here, okay? And then once you have your tracks, you have a bunch of tracks over here, okay? So you can just, you're probably not interested in all these bunch. You want to see one by one, okay? So you can just delete this ROI by just clicking on the ROI and just say delete. Do you see what I did? So I went back to the image. So I went back to the image over here, and I just removed the ROI that I had on my image. Have you guys done that? Yeah? So once you've done that, now you're safe, okay? So go on to add track processor. So in this add track processor, you have a series of functions over here, okay? So there are several things. You can paint your track, you can get ROI statistics of your track, you can motion profile your track, you can get intensity information of your track. But I will first start off with track processor ROI gate. Do you see track processor ROI gate in that area? Just click on that. So what this function does is basically it limits to show the tracks only in a ROI that you define, okay? So now if you like a particle, for example, I like this particle over here, let's say, okay? So if I want to see the track of that particle, all I do is I go here with the rectangle, choose a rectangle, and then draw a small, and then I see only one track displayed on my image. Is that correct? Can you guys do that? So choose any particle that you like. You say, I like that particle, I want to see the track for that particle, and draw a region of small region of interest in that particle area. The smaller it is, So this is how it is. Now what you can do is, once you've drawn once you've drawn this region of interest, some people have already come to the point where you can actually move this around, okay, and see the tracks of different particles. All right. So let's say I would like to see the information of this particle along. Okay. So just for example, I'm saying. So you go to a track that you like, and you say you stay on that track. Now you want to visually analyze whether the software has done a good job in the tracking. So what you do is you go and play your movie and see if the particle is actually moving on the track. Okay, or if it's some other particle on the track, you just keep watching it. See, in this case, in this track, I'm just going, it's still there. Do you see the particle moving on the track? Okay. At this point, however, because at this point I'm adding cyclosporin A, right? The particle jumps. So my track is lost at this point because there's a lot of motion. Okay. So what I do now is I draw a second region of interest on this particle over here. And you will see a second track appearing for the second segment of the particle tracks. See that? Okay. Have you guys identified these type of tracks? Once you've identified this, you can actually highlight these tracks. If you click on the track directly, you can highlight it, and then you click on this track, and you highlight these two tracks inside the track manager. Okay, so when you select the track on the image, you're also selecting the track inside your track manager. So you can just go ahead and select a track that you like. Okay, any track, it doesn't matter. Need to be. So once you've chosen your track, right, you don't want to use any of these other tracks. If you want, you can keep some of the tracks. For example, I'll just click on this and this. Maybe I'll keep these tracks, right? So I've chosen a bunch of tracks in this manner on my image, okay? And what I will do now is I will just go to edit, and I will say invert selection. So I'm working inside the track manager, right? So you go to edit and you go to invert selection. So when you do this, you are actually selecting all the tracks that you don't want to see. Okay? You're selecting tracks you don't want to see. So you can go ahead and once you say delete all these, select all the tracks that you don't want to see. Okay, delete links, delete selection. So now you're left out with only a few tracks that you are interested in analyzing. Okay? We are here now. So if you look at the track manager, if you just look at the screen for a moment, okay? So in time, what happens is that you see the cursor on my movie is at 170 time frame, okay? So you can locate the cursor by just moving in the time frame. You see this red line in your track window. So that tells you that is a location where you are at in your movie, okay? And you can see that these two tracks are actually split at this time, okay? You see, I mean, if you look at the screen, you will see that there are this red and this blue track, these two segments of tracks are actually split at this time. So you can actually take the last time of the track and join it with the first time of the second track, okay? By just clicking and drawing a line, right? And now you can go back to edit and say, organize track parent to child. So it's like father and mother, I mean father and son, whatever, right? And then you say fuse all track segments, then you get an entire track of that particle. Okay, so there is manual interpretation, right? So you first track the objects using the software, then you follow whether the tracking is good. Okay, so mostly it will do a good job tracking the object. At some point, when your particle jumps too far away, okay, in our case, when we add the drug, it just jumped off somewhere else, right? So then you want to identify where that particle moves. So you see the same particle there and say, okay, that track and this track belong together. Then you can join these two tracks to get the full tracks, okay? So now let's say you've got these tracks, right? So you want to do some analysis on these tracks. So you can actually, first let's look at visualizing these tracks in a different way. So go back to add track processor. And then there is something called as ROI gate, track processor ROI gate. Do you see that, everybody? Yeah? So you click on the ROI gate. No, we are already in ROI gate, sorry. That's not what you want. Time clip you want to go. Add track processor and go back to track processor time clip. Okay, did you find it? Did you find a time clip for track processor? Yeah. So now you say, it says number of detections to show before and after current detection. Do you see that? You can make that 3, 5, 10, just give a number to it, like let's say 10. So what this number tells you is that it tells you to show the tracks only 10 frames, okay, before and after. So you can actually click display future, so it'll also show where the track is going, okay. Now you play a movie, let's look at it. So this is another way to look at your tracks, right? Now you play a movie, you see that short segments of tracks are shown for your objects, okay? So this is another way when you have a crowded environment with plenty of tracks, you can actually follow a short segment of those tracks in those cases. You see how it works? Okay, so this is another way you can choose, okay, this particle is actually moving on the right track. So you know the track of the particle. Okay, once you got it, now we go to the real stuff, go back to add track processor, get to your intensity profile, track processor intensity profile. Okay, now you open that. So I'm just going to delete all the other tracks just for convenience. Invert selection. Okay. So once you open your intensity profile, if you scroll down, what happens is that, so yeah, I need to disable the track clipper. Okay, so make sure you disable the track clipper because it's going to show you intensity for only 10 frames if you have that on. So you have the intensity profile. Now you see the intensity profile. Can you see it? You have channel 0, channel 1, channel 2, and channel 3. Yeah? Do you see that? So this is the fluorescence intensity of your particle on your trajectory. Okay, as a particle is trafficking, it has the fluorescence intensity of green, which is not changing. You can see that the channel 0 is, I mean, in this particle which I'm showing over here, channel 0 is pretty stable. Channel 1 is your CPDS red. It's actually losing very slowly in this particle. And then at this point, we add cyclosporin A and you displace the CPDS red. Okay, so you do have some background fluorescence coming from your places where your particle is not there. So the way you remove your background fluorescence would be just go back up over here. So if you look at your intensity profile, below your intensity profile, there is something called as disk. Do you see that disk? You see a disk? Yeah. If you click on the disk, it tells you how do you want to show your fluorescence intensity. You want to show the fluorescence intensity within a disk of, let's say, 3 pixels, or you want to show a fluorescence intensity that is background corrected. So there's something called as disk background corrected. If you can choose that. So when you choose the disk background corrected, you get two options then. Okay. You get the diameter of the disk area in pixels. So basically that is the detection. So the first thing that you get, diameter of the disk area in pixels, is your detection diameter. Okay, it gives you a centroid of your spot. So it is automatically set to 2 and 4. I normally like to keep it to 3 and 9, right? So what it is doing is basically it's calculating around this disk of your centroid of 3 pixels. It's calculating what is the background, and then it's subtracting this background in your centroid, so you can actually get the real values. Okay. So this diameter for background I've set as 9. Okay. So now you can actually look at your. Look at your fluorescence. Now, if you look at your channel 1 fluorescence, okay, you can see that it goes to 0. See that? It actually goes to 0 fluorescence. Okay, so actually it's background. This is how you get your background corrected trajectory. Okay. Now you can simply export this information. If you keep going down, say export results. Okay, and you get a box that opens up. Export as what format? Excel. And then you say okay, and it asks you for a place. For example, I'll just put this back in the same data set. And you say, you call it whatever track name. But you please add that .xls extension to the end, because otherwise sometimes it does not save you an Excel file. As I say, did we all do it? You save the Excel file? Yeah? So now we can just close IC. And you can open your Excel file. Right? So if you open the Excel file, what you'll see. So in my case, I chose to use only one track, okay? Only one track. So because I deleted everything else, so now it shows me these tracks. So it gives you, this is the track name, so you can actually edit your track name. You can put the real name of your track on the track plugin when you're doing the track, right? And then it gives you a series of information here. If you look at the bottom, the tabs, right? It says channel 0 mean intensity, channel 0 max, channel 0 mean, min, median, and sum, right? And then if you keep going down, you'll see channel 1, same, all these information for channel 1, and then all the same thing will be repeated for channel 2 and then channel 3. Okay, so now you know that channel 1 is your CPDS red color, so you can just choose your fluorescence information, right? And then you can place it next to your fluorescence information of your green signal. Okay, then you can plot a graph in Excel. I think it's over here. And then you'll get a graph something like this of both overlaid colors. Okay, so here you can tell, you can actually normalize this graph by keep setting your first time point fluorescence as 100% and look at the extent of loss, and then you can actually tell that at this point I've added cyclosporin A and you've lost a CKD marker from this particle, and therefore this particle is a viral core that resides in the cytoplasm of the target cell. Okay, so that was the first exercise. So you can actually go back to the IC. Oh man. Okay, so if you go back to the IC program, you will see that this track manager add track processor plugin gives you a lot of other information also. It says there's something called as motion profiler. Click on the motion profiler, it'll open a different graph. It'll give you the trajectory, actual trajectory of your particle. This is how it moved. It started off here, you know, started off here, then it moved this way, and then it came all over here, and then it was just showing confined diffusion over here, okay? And then you can get your displacement values, your mean squared over here, right on top. See, you can tell you, okay, how many frames the track belongs to, how fast, and then there's something called a speed. So in synthesis, you can get a lot of other information about your track using the track manager plugin, right? Yeah, are we good? Yeah, so let's go on to do something more fun. We'll close this and okay. Okay. Okay, so now we'll move on to using a different plugin inside IC, and this plugin is called ECCLAM. Okay, and this is used for image correlation. Okay, so many biological insights can be obtained by combining fluorescence microscopy data sets with electron microscopy, cryo-electron microscopy. Okay, so when you put these two different, because fluorescence microscopy techniques can get you resolutions up to a few nanometers, like 10 nanometers when you do super resolution microscopy, right? When you do LiveCell imaging, you're limited to about 70 nanometers if you go in STED mode, okay? So you don't get the details of structures of whatever you're looking at inside cells. So what you want to do is you want to use a more powerful technique, which is a cryo-electron tomography technique, cryo-electron microscopy, to gain insights to the order of below 5 nanometer resolution. So you can start seeing the actual structures of what you're observing in your fluorescence image, right? So why is this important is because, you know, you can just taking this example of the virus, all these viral, like, you know, you have the capsid, the RNA, the matrix, the envelope, they're all very nicely organized within this roughly 120 nanometer viral object, right? But they're well organized inside this viral membrane. So you want to be able to distinguish where the matrix is and how this matrix shape changes with respect to capsid, how capsid shape changes with respect to the viral complex during the virus infection into cells, because these are really the key missing details of HIV-1 biology that we are finding at the moment, right? So this is a very crucial step that we would like to study. And this is an example of how you can go from a fluorescent image, you will see a punctate like that. Okay, and you cannot infer anything from this punctate apart from that there is a viral object right there, right? And then you can look at the object in more detail. You can start seeing some cones, and if you zoom in even further, then you can start seeing conical shape. Okay, and so that's how far you can go with to start seeing shapes and structures of viral complexes that are labeled fluorescently. That's what you can do with CLAM, right? So you can look at dynamics using fluorescent microscope, but you can actually look at full environmental context at high resolution detail using EM. At the very heart of correlative techniques, of this CLEM techniques, that is combining your fluorescence image with your EM image, is image correlation. Basically, you need to be able to figure out how you want to correlate fluorescent coordinates on an EM image. Okay, and that is the heart of the CLEM technique. So a simple CLEM workflow, which is published, with a collaboration with Greg's lab, published this work with Liz Wright, was that you first prepare grids. So on a fluorescence microscope, you look at the sample on a glass slide. When you want to go into a correlative EM, then you want to look at your sample on an EM grid. Okay, so you need to know how to prepare your grids. Then you culture the grids, culture your cells, or put your virus on these grids, and then you do a plunge freezing step over here. So this is very important when you want to go to cryo. So you want to be in a good situation where you have the right amount of ice thickness, so that your sample can appear and give you high resolution images of your viruses on the grids. Then you can do a fluorescence imaging after plunge freezing, or you can do a fluorescence imaging prior to plunge freezing. So fluorescence imaging will allow you to detect where your signals are distinctly to get coordinate information. And then you image them on the TEM, and then register do a image correlation between your fluorescence map and your TEM map, and then uses correlated location to identify your viral particles inside cells, and then collect a high resolution cryo-electron tomography data set over there. Okay, so cryo-EM grid, some of y'all might already know, looks something like this. There are various different sizes, different shapes, and they come on different dimensions. So normally for cells, what Liz lab found was that the gold grids are pretty inert, and they actually are useful to grow cells on them. And you have to take your grid, prepare your grid with cells or viruses, then you put them in one of these cryo plunger systems. In our lab, we use the CP3, and you set in your grid holder right over there, and there's liquid ethane. Just you blot your sample, remove all the excess water from your sample, and then you plunge it to get nice ice on your sample. Okay, so what we will do in this part of the workshop is we're going to see how we're going to correlate between a fluorescence image and an EM image, how we're going to align them to develop coordinates of where your viral particles are located. Okay, so data set that you have is it's a virus that is undergoing uncoating. These viruses are immobilized viruses, so they're stuck to the grid. So we have already taken a fluorescence image of these viruses before they were frozen, and then we froze them, and then we have a second image of the frozen grid. Okay, so we can open the IC software again, and I would suggest you go into the plugin tab. Right, so you go all the way down to the plugin over here, and you want to, if you have not yet downloaded the ECCLEM, go to the online plugin and simply type ECCLEM here, or CLEM, for example. So when you type that, it pops out these two. You see that? So you guys got it there? Yeah? So you simply say install. Just click on ECCLEM. Just click on it and say install. And after it's installed, just say apply and say okay. Then you'll have it on your plugin list. ECCLEM. So you can go to the data set that I sent you before. Okay. And if you go to the data set, where is it? So you want to first go into the data set that says low mag. Do you see there's a folder called low mag in the data set that I've sent you guys? No? You don't see a low mag data set? So what do you guys got? Oh, the ECCLEM data set was not distributed. sorry. Okay, so I will show you. Sorry. So for example, all right, so this is, I don't know that I didn't send it across. Okay, so I'm just going to walk you through this, all right? So this is how a fluorescence data set will look like. This is a grid that we took a fluorescence image on. Okay, let's look at it, how it actually looks. So, right. Maybe it's just the presentation. Okay. So sorry that I have not sent you guys the data set. Okay, so this is how the grid with the bound viruses will look like. You have a bunch of broken squares, then you have a bunch of red spots. All these red spots are beads, fluorescent beads, that we can use in both the fluorescence image and EM images. Okay, and then when you do, you can actually, the way you acquire the whole grid image is just take the tiles of this image, and then you get the whole grid image under 40x objective, right? And then you can take a high-res image of a particular region of interest, in this case, this area. You see all these green dots, these are all viral complexes, and then you have these red beads, which are 200 nanometer beads, and the big bead that you see here is a 4 micron bead, okay? So this allows for image correlation. So now, if you go under the, now you've taken this and you've frozen it, right? If you go under the EM, then you get an image of the grid something like, that looks something like this. Okay. Now I would like you to try to figure out where is number 1. Because it's like, so I can tell you that the center is out here, and it's a square close to the center, right? So all you have to locate is a center in this image. So this could be the center. Yeah, you say this could be a center. Then you see a broken square over here that will correlate with this broken square over here. Then you have a second broken square over here, diagonal to that. That will be about this. So in between these two broken squares will be this particular square where you have this big bead and a small bead, and those two beads are there. Okay. So by eye, we are doing a correlation like, okay, we can say there are broken squares here, and then you have to also look at, after this, you say there's a broken square here, intact square there, broken, intact, and then 2 broken. You can look at that here, right? Broken, intact, 2 broken, right? So you can be almost certain that this is a square that you want, that you are interested in collecting your till series data set, okay? So now you go into this square. You can actually do all of these automatically on EasyClam, okay, which is unfortunately I did not give you the data set. So you can go on these on EasyClam, and now you can look at the high resolution image alone. So here you have a high res image of the beads and the virus on cryo-EM. They will appear something like this. You have dark spots and big blobs over here, and this is your fluorescence image, okay? So you have EasyClam, this plugin. If you just open the plugin, at least you will go through the.
How does this scale in the pixel density?
So it does a nonlinear transformation. So at the end of the day, right? So you need to have your EM images should be on proper scale. You need to have the information of your pixel sizes of your EM image, and then you need to have the pixel sizes of your fluorescence image. This is what you get from the metadata, right? So you put it in the Clem software. There are basically 2 selections over here. One, you say it's a fluorescence image you put there, an electron microscopy image you put here, and then you start saying play, right? So once you say play, you come to something like this. It gives you some information about how to proceed. It actually gives you the protocol on how to proceed with this. And you start with looking for features that are identical on both. In this case, I see these features. There are 3 beads out here and 3 beads out here, and it's about, so then you start by adding points one by one. Okay, so you stay, this is my first bead, then you identify the same bead on the second, on the fluorescence image. You add, let's say this is my point one, so you match them both on both beads. So you do that for 3 beads or 4 beads, 4 locations that you can be certain of on both of these images, and then you just press play, and then it will give you a transform image like this. And for example, yeah, sorry, so it'll give you a transform image something like this, where you have overlaid your EM image with your fluorescence image over here. Okay, so for example, here these are 2 beads. You could not have told that on a fluorescence microscope, but on the EM microscope you can actually tell. So you can actually say these are 2 beads on the EM microscope. That something like that you'll see over here. You see. So you have, once you overlay the images, your beads will appear dark in color. So you can make sure that these are overlaid images. And now you can actually open your spot detector plugin, right? And yeah, sorry. And then you can detect the spots in your overlaid image, which will give you an Excel output, which you can save outside. And the Excel output will have the coordinates of all your viral particles, where they are located. You take these coordinates and you put them inside the EM software to mark the points where you are interested to collect your TIL series data set. Okay, so I'm very sorry that I did not give you the data set. I thought I did send it to you guys. So, but that's how it works. All right. So if you want, I could give you the data set right now, and we can play with it a little bit. Yeah. Interesting. Yeah. Let's do that. Yeah.
Or maybe we should sit around good meetings. So the people back here can't see it, and we're recording it in real time.
Just going to. That's terrible. I can also send it by email, I suppose. So. So if you have the data sets, let's go to easy client, right? So you want to go for the no, low res. So let's open both the low res. What's the low res data set that you have there? Okay, so you have both your fluorescence and your EM image, right? So now you go to EasyClam. Okay, so it tells you add points to the 3D on target image only, and then it tells you drag the point in source and right click, and then you can move those points across the thing. So what you will first do is you select the image that will be transformed and resized like fluorescence image. You see that on EasyClam? And you say that is the stitch NA file. Okay, that's a stitch NA file. All right, then the EM image is a wild type IP file, WT IP file. You get it? Yeah? You chose your files. It can be either or. You can put one as a fluorescence image, the other one is a EM image. And then you say play. So when you say play, you will see something appears on the screen of your images. Do you see something like that? Like a red, something that says in red? You guys see that? Something that reads in red?